Hi, good morning, everybody. Today we'll be talking about the next chapter called nutrients. Now, for this particular chapter, right, the only difference between students doing O-level pure biology with the science biology in the N and O-level is that there is this segment of the chemical compositions of the nutrients. This is only required for students doing the pure biology in O-level, while students in doing science bio in N and O level, you don't really need to know this. Okay, and this portion about the chemical composition tends to be tested in the MCQ for the pure bio section. Okay, so let's begin. So for our lesson layout for today is we will be actually understanding the need for food. Why do we actually take in the nutrients? So you realize it's mainly for maybe there will be two segments. So we will be asking like why animals or humans actually need the food, and the other one is plants. So both of them will have different, more or less the same yet different kinds of ways to actually obtain nutrients, and at the same time, the reasons why. We'll be touching about water. Why water? Water is actually one of the main components in your human body. So in this case, right, we actually need water to run a lot of our chemical reaction. So be expected to ask essay questions related to like what are some importance of water, or what are some functions of water, if ever this comes as an essay question. Carbohydrate, fats, and proteins, these are the other nutritional groups in which you will need to focus more on the food test. So if you have seen the past few years, science bio practical examinations, they do ask most probably two food tests to be conducted for the science bio O level. For pure bio O level, because of the recent change in examination format for practical, it's quite hard to see any trend. And what happens is, how do we then test in the paper and pen, like in your paper two exam papers, is that they would actually ask for the observation, or they would have given you the results, and you're expected to infer what are the substrates, or what are the nutrient groups that are actually present. And this is usually relatable to other chapters, like the chapters of enzyme or the chapters in animal nutrients. So along the way, I will discuss more and we'll see. Okay, so without any hesitation, let's begin. So first of all, you must be able to explain why organisms need food. This means giving reasons and of course type of nutrients. And in this case, we have studied quite the three major groups: protein, carbohydrates, and fats. But there are also other nutritional groups like vitamins and mineral salts that don't really encompass us in O level and N level. Okay, so for now, just focus more on glucose. Yeah, sorry, not glucose, but your carbohydrates, your protein, and also your fats. Water can be considered as one, so maybe you put water. Okay, but actually water I wouldn't really classify it as a form of nutrients. I would say it's a biological solvent. So the reason why we need food, right, generally is to provide energy. Provide energy for what kind of process? So for humans, we usually need to move around, so we need energy. Some of your body organs, right, like the heart, let's say the heart needs to be continuously pumping. So this also requires energy. And if you remember the chapter on cells, you realize that the organelle that actually is responsible for this is actually your mitochondria. So what happens is mitochondria would utilize the uh, glucose, or what we call the carbohydrates, and then you are able to then release energy, and these energies will then be used by your cells to do its functions like moving around, respiration, etc. Okay, let me write it here. Respiration. Next, to supply raw materials to make protoplasm, what this means is mainly for your cells to undergo repair. So when you use your cells over a long period of time, right, it generates heat. And what heat actually does is heat will result in wear and tear. So imagine when your heart is beating, you are also indirectly creating energy. You may not feel that there's a lot of heat, but to the cells level, you will not know what is the threshold of heat that they can take. So over time, there will be friction generated, and then heat can be generated, and all this will actually result to your cell's structure to be less stable. And what happens is cell needs to continuously repair these structures so that it will be able to be survive or to be healthy. And where do then get all this information from? They get it from the nucleus. So if you remember in the chapter of cells, right, the nucleus is this database of all the genetic structures that makes up the cell. So what happens is the whole process of taking the genetic information to convert it into a protein and then into a structure of the cell. All this is the mechanism of the cell construction. And in this case, right, we do not just create things out of nowhere, even though we have the information. But if I do not have the materials, I will not be able to create a product. So where do I get the uh, materials from the food we eat? Why? Because if you break down even to our human body, the atoms and the molecules, right, you realize that we are mainly made up of carbon atoms, hydrogen atoms, oxygen atoms, and the only way we can get all this is through food. We are not able to undergo photosynthesis because we do not have the organelle chloroplast to be able to trap light energy to convert it into food nutrients. Okay, so and of course to help us keep healthy. So these are the three nutrients: carbohydrates, fats, and proteins. Okay, so for proteins, there is actually online. You can go and look for the amino acid table. There are quite a few of it, and then just imagine that proteins are these molecules or particles of amino acids that are joined together, and they are not usually in a straight line or linear, we call it. They are usually clump up together. So imagine if you take a ball of string, you have a string, and then you coil up into a structure. So this is what we call like a protein. So usually it's a amino acid structure, amino acid that will slowly lead into a polypeptide. Okay, this one we will talk more about it. In JC, you will learn the different folding effects, the different primary, secondary, tertiary structure of a protein. But in O level and N level, this is not required. Fats are mainly made up of certain other molecules that are actually uh, which I will talk about in the later part. I don't want to confuse you here. Carbohydrates examples are like your starch, okay, your glucose. Then if you have watched my video previously in movement of substance, they will use sucrose. These are examples of carbohydrate. So some of you may ask, what about sugar? So sugar is the layman's word for carbohydrate. But in exam, please do not use the word sugar. Sugar shouldn't be used in O level and N level. Instead, use words like starch, glucose, sucrose, as these are more specific. Sugar is a term that usually, if you write maybe in primary school, they will be accepted. Okay. So we look at water now. So for water, mainly you need to understand the roles of water, meaning how can water perform its function to living organism. So first of all, in order to understand the function, we take a look at some of its basic structure. So what is water? So water is a solvent. It will be good if you can use the word biological solvent. So biological solvent, the meaning of biological here means that it is a solvent usually used in a living organism body. Okay, so that's biological. In which chemical reactions takes place. Chemical reaction can be anything. For example, when you actually, if you remember your chapter two of your sorry, not chapter two, when you were in sec two, you learn the chapter um, changes in metal. So you will see that there's chemical changes like oxidation, combination, not chemical oxidation and other processes. respiration. So these are examples of chemical reactions that can happen in your human body. And what is the purpose of all this? These chemical reactions can lead to you repairing the cell, lead you to actually creating something new. It can even be just a reaction to release energy. So there are a lot of general chemical reactions. Then it is essential. The meaning of essential means important. Okay, I will try my best to explain some of the jargon words due to the audience. And through this, right, it having an in being an important component of what your cells, tissue, digestive juice. Digestive juice usually contains enzyme, which we will be covering in the next chapter, and of course the human blood. It helps to control your body temperature. So if you look at some those gaming PC, right, they would have all those hydraulic system, or if you have a very expensive e-scooter, you also realize that it has this they use the hydraulic brake, but sometimes over from my understanding is there is also some form of cooling system to help to reduce the brakes. So why is it so? Because in Singapore it's tentatively quite hot, and by if you do not cool your brakes, right, what will happen? It will generate even more wear and tear, and then every few maybe days or weeks you would have to actually change your tires and things like this. This happens almost everywhere. So even in human body, right, we have so many organs actually exercising, working, and generating heat. So there must be a cooling system that help us to manage or to maintain the temperature within the body, because you will realize that in the next chapter that I'll be teaching is that enzymes, they work on a very narrow range of uh, temperature with optimally. So in that sense, right, we need to ensure that our body conditions are usually not influenced by external factors, and it's usually maintained at its peak condition. And this is why we in humans we are also warm-blooded, to such that the external environment temperature do not actually heat up our body, as compared to maybe a cold-blooded creature which is influenced by external temperature, and you realize that they tend to have no way of controlling their body's temperature. Okay, so that's one example. Next is to help to transport dissolved substances. So it's like, let's say your cells in the leg create some nutrients. You would want to then transport to maybe to the brain for some use of the nutrients, or maybe the food you eat is being absorbed by in your small intestine, and then you want to bring this absorbed nutrients to all parts of the body. You will need water. Why water? Because water is very fluid. It moves fast. If you look at if you compare different kinds of solvent, right, like oil, when you pour it out, the flow will be quite slow as compared to water, which is very, very fast. So, and not just the flow of the water, but how the whole molecules within, like your nutrients group within with water, move around your body. So, water is one such area. So what about plants then? So water is important for photosynthesis. We will talk about this in the next few chapters, where you will see that water is basically one of the key components for photosynthesis to occur. This one we already covered the term. So it helps the plants to actually become turgid so that it stands upright, and so you can get maximum amount of light energy. So this one would then help you for your photosynthesis. Then it is needed to transport mineral salts up the xylem. So where you usually get it from, the mineral salts from the roots via active transport. Okay, we'll talk more in the next few chapters as well. So you'll start to see that actually all the chapters will start to link to each other, so that it paints a very nice story. So once you have already absorbed the mineral salts from the soil via the roots via active transport, at the bottom part of let's say a tree, it needs to bring all these mineral salt against the gravity up to the plant. So definitely you will need water to help you there. And of course, while it moves from the roots to the top of the tree, so from bottom to top, This other one is from the leaf, right? So the leaf is where photosynthesis occurs. So over at photosynthesis, you also create glucose. So this is from the top to the bottom of the leaf. I spell bottom short form, so yeah, but BTN means bottom. So what happened is you will actually see that this is mainly a transport kind of system. Then of course, how much water do we need? It's basically the same idea with how much water are you losing, because but in our body we tend to actually we need water, but then how, so usually the rule of thumb is maybe 8 glass of water per day, but you ask yourself, do you lose 8 glass of water per day? Let's say you sit in an aircon room, you don't really sweat. Does that mean that you need to drink 8 glass of water? I mean, of course, for healthy reasons you do. Then you just go to the toilet more often to lose that water. Then some of you will ask, oh, if the recommended is 8, but I drink 2 cups, do I die? No. It's back to then. Just think of it this way: your body is one closed system where it's like one cup, one container. This container, right, has a limit. Too much water, it will overflow. Too little water also is an issue. So it's about the equilibrium. So as long as your volume of water intake is more or less similar to your volume of water outgo, right, then I would say that you are in that's the amount of water that you really need. And everyone has different body size, everyone has different physical activity. So there is no hard rule, a number per se to how much water do you need. But if you understand that it really depends on how active the person is, how healthy the person is, and of course the environmental factor, aircon room, stand under the hot sun, in which we lose water via sweating, urinating, and of course breathing. So when you breathe, right, I mean, you can try now. If you put your hand there and you breathe towards your hand, you'll feel this it's a bit damp. Okay, it's not because so much as Singapore is very humid, that's why you feel that sweaty palm and things like this, but because through your breath out, right, you actually, there is some water there, water droplets there that gives you that particular feeling. And this one we'll talk more in the next few chapters as well. Okay, so next we come to carbohydrates. So carbohydrates, okay, for this segment you need to know the chemical elements. This one is mainly for students doing pure biology. 66093 in O level. For the students taking science bio and combined science bio in O and N level, not really tested, but it can come out as MCQ for science bio in O level. Okay, so that next is the understand that glycogen is actually made from glucose as well. Investigate. So this is important. Investigate for the present means you must remember the food test. Now, in practical, for science bio O level, they do actually give quite detailed steps. But at the same time, there are some common steps that you need to know. For example, how to conduct a food test or the test for reducing sugar, you will need to know. These are steps that the exams may or may not give you, and most of the time they don't really give it to you because you are expected to actually know. Volume wise, you would also have to have a rough idea how much volume it is. For pure bio students, this is non-negotiable. You have to remember it. Okay, so next, so what are the compounds? The atoms, the elements that makes up carbohydrates. They are just three: carbon, hydrogen, and oxygen. If you look at your periodic table, carbon is a capital C, hydrogen is a capital H, oxygen is a capital O. Okay, and they follow this formula. So only carbohydrate follows a fixed formula, which is, you see, CnH2MOM. Now, what is this? N represents the number of carbon atoms. Atoms are not molecules. M here represents the number of oxygen atoms. Now, over here is oxygen atoms. In this sense, right, it's not the O2, it's just O. You count the number of O. Then, of course, the M here is the number of hydrogen atoms in total is just 2 times the number of oxygen atoms. That's why it's usually M and M for only carbohydrate. So do take note, these are which are the common numbers and which are not the common numbers. So this is something, and this is the key thing. This usually comes out as a statement in your MCQ to separate what is actually a carbohydrate and what is a fat. Because for fats, they don't really fulfill this ratio. Carbohydrates they do, so do remember this. And this ratio is what? Is basically the ratio of hydrogen, which represents in the 2, and hydrogen, and 1 is the oxygen atoms. So it's a O and a H. Yeah. So these are the different. So when you look at carbohydrates, if you want to break it down into multiple tiers, how is it going to be like? So monosaccharides are the basic simple structures of sugar, followed by disaccharide, which is the second part, and then third is the polysaccharide. So for monosaccharide, you have your glucose, fructose, galactose. These are the examples. Don't worry about memorizing it now because if you revise this, and especially if you watch this video again and again and again, I can tell all this will start to will become more familiar to you. If I won't expect you to after you just watch it once, so then you remember. That's why if you can just remember to subscribe, like, and click the notification button so that you could just rewatch, and then hopefully it will help you because to do well in your examinations, the basic fundamental that you need to know is the content. Why? Because there are some questions. So in exams, right, there are two types of questions. Questions that test on your memory, and the other one is to test for your application. So what will happen is if let's say I ask you, oh, carbohydrates is made up of how many types of groups? This kind of answer is whether it's a matter of you know or you don't know. It's not about how you decipher and then apply. Okay, that's what we meant by that's the main purpose of these videos, in which right it's to actually condense lessons that takes about 2-3 weeks into something that is within 1 hour or lesser for you to do your revision. So think of it this way. I basically what happens is from you are basically trying to compress and cram everything up. That's why it's called crash course. Okay, and along the way we will also be discussing what are some common exam techniques that you may want to identify and take notice of. And when you do your 10-year series, you'll realize that the things that coming out is actually quite important and it's quite repetitive. Yeah. Okay. So while there's so many things to study, so many things to memorize, I would suggest that if you cannot memorize so much, at least the mere minimum is to remember the basic key things. Okay. And of course, monosaccharide, if you look at the word mono, if you look at the word mono here, mono means one. That's why it's single. Di means two, double. Poly means complex. So this is how we usually represent molecule size in biology or even in chemistry when you do organic chemistry. So all monosaccharide and disaccharide except sucrose are reducing sugar. Okay, this term reducing sugar, as of now, you don't have to worry so much. Um, just take it that it's a type of reducing sugar. In your exam, they don't really test you on non-reducing sugar. So just assume that it's reducing sugar for all. Okay, so how are disaccharide formed is there is a condensation reaction. So if you remember condensation, like in chemistry, like they say, oh, your cup of ice water is undergoing condensation. Then you see water droplets on the surface of the cup. So what you can see is that previously there's no water droplets, right? That's why you don't say that, oh, the cup undergoes condensation. It's only after the formation of water droplets on the surface of the cup that say, oh, there's condensation. So what this actually also tell you is that condensation is the process whereby you actually produce water. So how is the water produced then? The water must come from somewhere because you are not an alchemist, you suddenly conjure up water. It comes from the molecules of the nutrients. And the molecules of the nutrients, right, they actually join together to form a larger molecule. And this is true because there's the removal of one molecule of water, therefore water is released, and therefore you can see the water droplets. Okay, and what actually this means is that condensation, do you see? It helps to join. So what happens is, if you look at the molecule here, there is maybe a hydrogen bond here and maybe a hydroxide bond, which is a OH bond. So what happens is, when before they are combined together, right, they actually have this structure. But when they start to join together, there is no space for these two molecules. So water is removed in the form of H2O. Okay. If I have time, I'll do some animations of this in another video, but this is what basically condensation reaction is. What about hydrolysis? Hydrolysis, on the other hand, is the opposite of um, condensation, where one produces water, the other for hydrolysis means you use water. That's why you use water with the help of enzymes, like for example, maltase is an enzyme, to help to break this bond, And by breaking this bond, you have empty spaces over here. So what do you do with the empty spaces? You add the molecules of water. So it will actually join back, maybe one hydrogen here, maybe another hydroxide here. But of course, there's more atoms here. This one is too complex for you to understand at O level and N level. You will see more in actually A levels. Okay, so this is how it's actually done. And usually for hydrolysis, it usually requires enzymes and not just water alone in your human body. So how do we test for reducing sugar? Usually we use a Benedict solution test. So Benedict solution test, just remember everything must be in twos. For rule of thumb, heat the contents in a boiling water bath. This is a very key important word. If you never write the word boiling right, you'll be marked wrong. If you never write the word water bath, you'll also be marked wrong, and it's not throwing it into boiling water. It's actually into a boiling water bath. So what is a boiling water bath? How is it different from heating? Now, boiling water bath means that within a beaker, there's already a boiled water, and you put your test tube. So boiling water. And maybe heat is here. This is a boiling water bath. And make sure when you do your practical, whatever contents that is in the test tube here has to be lower than the water level in the beaker. This helps to ensure that all the contents are actually undergoing boiling in the water bath. Now, one common mistake that I see is that a lot of students, right, they tend to fill up their contents all the way to the brim, such that this region here is above the water. So you can see, so this is actually not a very good, this will actually result in your experiment to have very different observation. Why? Because this area here will not react, or react very slowly due to it not being exposed to the full water. So what is heating? Heating is just basically you heat the contents like this directly. Now, of course, boiling water means what? You throw all the, so boiling water, if you just say boiling water, that means without the test tube, you just throw all your food substances inside the boiling water. That's why we usually use the word boiling water bath. And how do we do it? We usually do it at 2 to 3 minutes. Anything more than that, right, you will actually see that there will also be a change in color. So I would say a very fair gauge is within 2 to 4 or 5 minutes, you will see the color change immediately. But if you boil anything more than that, right, you will see it turn blue, brick red. Reason being is because of the Benedict solution. Too long, Benedict solution can also undergo decomposition, and that will give you the color observation. Then it will give you a false test. Okay, so try to remember the magic number 222, and then another one is to shake. This is a crucial thing. If you don't write to shake, you are just basically adding the contents, then you have two layers. So maybe you have your food here, and maybe you have your Benedict solution. So when you do that, what will happen is you will when you do the experiment, you will see that there's two distinct layers of color. One will be blue color, and maybe your food will be the one, if there is reducing sugar, will be brick red. So this is the experiment observation you will see. But if you shake, everything is equally mixed. So you have the food plus the Benedict solution. Then you will see a continuous change in the coloration, and not only that, you will see the precipitate form. Okay, so these are some basic steps that you need to know, the reasons why you are doing it, so that in the event the structured question asks you why are you doing these steps, you're able to explain. If practical asks you this kind of question, you know how to explain. Okay. So what will happen is you will get either these few colors, green, yellow, or brick red precipitate. Now all these will indicate different amounts of sugar concentration, and don't use the word amount. Amount is too generic. I will usually tell my students to use either the concentration, use words like concentration, um, use words like volume, amount of sucrose, amount of sugar, amount of heat is too generic. If you say light, amount of light, why not you say light intensity? So these are some ways you can improve your quality of your answer and impress the marker. Okay, and of course to get higher marks. So this means that traces means very little, so this is for trace. Yellow means moderate, means like somewhere in the middle, and of course brick red is large amount or large concentration. Okay, don't use the word volume, just say large concentration. If you want to use large amount, you can also do it. Depends on the school whether they accept it or not. Okay, so yeah. So there are many different types of complex sugars, carbohydrates. There's a starch. So now we are talking polysaccharides already. Polysaccharides already. So if you remember, there is the monosaccharide, disaccharide, and polysaccharide. Polysaccharide is only in this area. You have only three to remember: starch, glycogen, and cellulose. Cellulose, if you remember, is actually the main component within a cell, the cell wall. And the cell wall, what it does is it helps to protect the cell. So this one is quite straightforward. Glycogen and starch. No starch, right? One common mistake is that in primary school we always learn that to test for photosynthesis we do a starch test. Now do take note that photosynthesis do not directly create starch. Instead, what happens is photosynthesis will actually create glucose first. Is this excess glucose, then it will be converted and stored as starch. Okay, then it's because of how you test for starch, then it tells you whether photosynthesis occurs or not. But you can also use a reducing sugar benedict solution test to test for photosynthesis. Just that it's not that accurate because glucose is just it's too freely available everywhere. So when it's too freely available everywhere, it doesn't give you a very good indicator whether photosynthesis has occurred, but it can tell you that with some level of assurance that yes, it happens. Glycogen, on the other hand, is stored glucose. Okay, this one you'll also learn more in the next chapter and the next few chapters in animal nutrients. What happens or nutrition in humans? Okay, so what happens is glucose by itself is too small and it's too easily used. So in a sense that when we burn, right, we burn very fast. Then at times when we do fasting or what's not, when we need glucose, then where do we get it? So we actually most of your excess glucose are then converted to glycogen by your liver. In the sense that it helps to store such carbohydrates in times of when you need it. Okay, so and why store in glycogen? Because glycogen has certain properties that allows the storage to be easy. Like it doesn't influence your water potential in your body. Because you see, right, your blood, right, in your blood there's a lot of cells, like a red blood cell and what's not. So imagine if you adjust the concentration of your blood, right, by drinking a lot of water, then you make it very diluted. Your blood become very diluted. So more water will enter into your red blood cell. But at the same time, if you don't drink a lot of water, your red blood cells will get created. So this whole internal structure, right, of your body, right, is being managed by some other by other system, feedback system, which is in the chapter of homeostasis. So for now, you just need to know for glycogen, it is just a molecule that allows easy storage. And the hydrolysis of starch is by maltase, sorry, not maltase, amylase. So amylase breaks down starch into maltose. How do we write it down? We usually write the starting material, followed by an arrow, followed by the molecule that it forms. In this case, it forms maltose. So the name of the enzyme, we usually write it at the top. Do remember, how do you recognize which is a molecule and which is an enzyme? Usually enzyme ends with a ASE. See, like maltase, this is an enzyme, so it will be maltose. It will form to a glucose, and this is by maltase. And of course, glucose is being the smallest one, it will not be able to, a monosaccharide, it will not be able to break down even further. And do take note, most of the time, your enzyme names, right, it ends with ASE, and also the first part of the name follows the previous molecule. So it's easier to remember for some of you. Okay. So how do we test for starch? Very straightforward. You just use iodine test. You take 2 cm³ of the food sample, put it on maybe a white towel, and then just drop a few drops of iodine solution. I know some of you like to spray a lot. Do not spray a lot. You spray a lot, right? You'll be whole black. The whole thing will be dark. Then you cannot see anything. The color of iodine, right, is usually yellowish or brown in color. So if there is starch present, you'll form blue-black solution. If starch is absent, okay, the solution remains brown. So how should you write your observation in your practical exam? You always say initial observation change to a final observation. So you say like, okay, after adding iodine solution, the solution, uh, the brown solution changes to a blue-black solution. This indicates that starch is present. You do not just say a blue-black solution is formed. Similarly, if you look at the Benedict solution test, okay, so of course, oh yes, so if reducing sugar is absent, if reducing sugar is absent, okay, solution remains blue. Okay, so these are some of the things, and do take note, you need to use the word PPT for here. Okay, and then yeah, finally we look at the functions of carbohydrates. Carbohydrates are needed to provide energy for cell's activity to support your structures like a cell wall, for conversion into other organic compounds like amino acid and fats. So generally these are some, there are, of course, all these are just functions, you just have to memorize it. You can see there's a lot. So for essay, right, generally you don't have to remember so much. Uh, if it ever asks you, just remember 2 or 3 will do. And some of these things, right, as I say, you go along, if you revise and watch this video a few more times, you'll realize that this will come in naturally to you. Okay, so these are the functions of carbohydrates, and do stop the video here if you are interested. If it's too long for you. So today we already just covered the role of water, the role of carbohydrates. In the next video, in part 2, I'll be actually sharing more with the other substance, which is fats and protein. Okay, so if you're interested, just pause the video. I'm now going to start to break down the videos, right, to make it shorter, so that at least you have bite size to know. Okay, then of course watch the relevant one. Okay, that's all. And remember to like, subscribe to my channel, and also if you can share with your friends, such that everyone could actually learn and grow together. Okay, thank you. Bye bye.